Hello and welcome to SAM online class. This is Dr. Subrat Kumar Mohanty. Today we shall discuss about recombinant DNA technology. The technology that is used for the construction and manipulation of recombinant DNA is known as recombinant DNA technology. Recombinant technology involves the use of several techniques and procedures for the construction and manipulation of recombinant DNA. What is recombinant DNA? The DNA that is composed of sequences from multiple sources is known as recombinant DNA. Recombinant DNA technology is also known as genetic engineering. This technology has several applications. For example, recombinant DNA technology is useful for the production of genetically modified organisms, for example, transgenic plant. It is also used for commercial production of several proteins like insulin, human growth hormones and other. It is also used for cloning. Then, what are the tools of recombinant DNA technology? The main tools of recombinant DNA technology are different type of enzymes. Second tool is vector. Third is host. These are the three main tools of recombinant DNA technology. One is enzymes, second is vector, third is host. First is enzymes. You know that enzymes are the biocatalysts that increase the rate of biochemical reactions. So several enzymes are used in recombinant DNA technology. Various enzymes that are useful are restriction endonuclease, ligase, DNA polymerase, reverse transcriptase, polynucleotide kinase, terminal transferase, exonuclease 3, bacteriophage lambda exonuclease and alkaline phosphatase. These are various enzymes that are used for various purposes in recombinant DNA technology. The second tool is vector. What is vector? Vector is a DNA molecule that help in the transfer and integration of DNA. Several types of vector are used in recombinant DNA technology. For example, plasmid, cosmid, bacteriophage vectors, different type of artificial chromosomes. All these vectors help in transfer and integration of DNA. Vectors can be divided into two different groups. One is cloning vector, other is expression vector. Cloning vector is useful for cloning, whereas expression vector is useful to synthesize different type of proteins by using the principle of recombinant DNA technology. For example, by using expression vector, we can synthesize insulin within E. coli. Third tool is host. What is host? Host is an organism into which the recombinant DNA is introduced for replication and for expression. Most frequently, E. coli is used as the host in recombinant DNA technology. So now we shall discuss about different type of enzymes that are useful in rDNA technology. First enzyme is restriction endonuclease. Endonucleases are the enzyme that produce internal cuts. And restriction endonucleases cleave the DNA into fragments. They cleave phosphodiester bond at or near specific recognition site known as restriction site. To do so, The restriction endonuclease cleave two phosphodiester bond. One phosphodiester bond is cleaved in one strand and the other phosphodiester bond is cleaved in the complementary strand. So restriction endonucleases are very useful in molecular biology and in recombinant DNA technology to cleave DNA molecules at specific position. The 1978 Nobel Prize for Physiology or Medicine was awarded to Warner Arbor, Daniel Nathans and Hamilton Smith for the discovery and characterization of restriction endonuclease. Over 3,600 restriction endonucleases have already been identified. Out of them, 800 restriction endonucleases are available commercially. Restriction endonucleases are present in bacteria and archaea, where they protect the microorganism from the invading virus. Restriction endonucleases are classified into 5 different types on the basis of composition, cofactor requirement, nature of target sequence and position of the recognition site relative to the cleavage site. The various types of restriction endonucleases are type 1, type 2, type 3, type 4 and type 5. Out of 5 types of restriction endonuclease, type 2 restriction endonuclease is used in recombinant DNA technology for cleaving phosphodiester bond within or near specific recognition site. The recognition site that is recognized by restriction endonuclease is known as restriction site. Type 2 restriction endonucleases are the enzyme that recognize and cleave at the same site. Type 2 restriction endonucleases form homodimers. They recognize and cleave at the same site. The recognition sequences are undivided and palindromic. The sequences contain 4 to 8 nucleotides. Next is type 2 restriction endonucleases that do not require ATP or S-adenosyl methionine for their activity. However, they require divalent magnesium ion for their activity. Restriction endonucleases cleave both the strands of a double-stranded DNA molecule. Some of the restriction endonucleases produce staggered cuts, whereas other restriction endonucleases produce straight cuts. So when a restriction endonuclease produces staggered cuts, that means these two strands will be cleaved at different position. This strand is cleaved at this position, where the complementary strand is cleaved at another position. So due to such type of staggered cuts, the ends which are produced are called as sticky ends, because these two strands are cleaved at a different position. So there is a single stranded protruding end. This is the single stranded protruding end. This end is called as sticky ends or cohesive ends. Such type of ends are useful in annealing two DNA molecules. Some other restriction endonucleases produce straight cuts. When they produce straight cuts, both the strands are cleaved just opposite to each other. At the same position, these two strands are cleaved. So, such type of straight cuts produce blunt ends. Such type of ends in which both the strands end at the same point is known as blunt ends. This is about different type of restriction endonucleases.